We looked previously at the glycosidic linkage, which connects monomer units of the carbohydrates, so glucose, galactose, fructose, into disaccharides and polysaccharides. But once we have those larger carbohydrate structures, we also need to break them down into monomers like glucose, individual carbohydrate rings, as is shown on the right of the screen where we see 2 glucose monomers. This is necessary because in order to continue our metabolism, we need to have a monomer unit that we're going to break down into what eventually forms our energy. So larger carbohydrates must first undergo hydrolysis in order to be these usable glucose units that will begin what happens next, which we'll talk about a little bit later. And so in order to do this, we need to cleave the glycosidic linkage, which is shown here. And what we're going to see is that not all glycosidic linkages are created the same. This is a quote-unquote easy 1,4 alpha glycosidic linkage named based on the carbons that it connects, carbon 1 from the left monosaccharide and carbon 4 on the right monosaccharide. And it's an alpha type glycosidic linkage, meaning that both of the linkages are kind of going down and keeping things in a certain conformation, which we'll look at that a little bit later as well. When we hydrolysis, this sticks a water into the maltose, which is the disaccharide shown here, and leaves us with 2 glucose monomers adding in the water. The OH from the water goes to one of these monomers and the H goes to the other. The oxygen that was part of the glycosidic linkage will still be part of that unit. This again is that first step in breaking down a larger carbohydrate to begin the process that happens next with a glucose. What about if we change this, right? What about lactose? So lactose is very similar, except instead of having that easy alpha bond, we have this beta bond. And the lactose disaccharide needs a special enzyme called lactase. And we'll talk about enzymes later, but that's what's in charge of breaking this down. These don't just break apart on their own. You need other biochemicals to help break them apart. If lactase is missing, this is not really possible to break apart, and then that can cause, cramps, diarrhea, things like that. Lactose intolerance means unable to handle lactose, which is this disaccharide, this sugar. The key here is that compared to maltose, lactose has a beta glycosidic bond, and this requires special treatment in order to separate. So again, the alpha bond versus the beta bond causing pretty big differences in our structure. And I apologize, I think I said they're both glucose, galactose, and glucose for the lactose disaccharide. Cellulose, look at this again, right? We have 1,4 beta glycosidic linkages for cellulose. And this one, we just don't have it at all. We have lactose, or the lactose consuming enzyme if we're not lactose intolerant, but no human can consume cellulose. These 1,4 beta glycosidic linkages, when they're part of this larger structure, just cannot be broken down. But animals that primarily eat grass, they can break down the cellulose. They can digest the cellulose, right? Humans cannot, but other animals have developed tools to get food out of this source that we cannot get food out of. Cellulose is a carbohydrate that's in the cell walls of plants. It gives rigidity to wood, plant stems, grass. It's a very important biological molecule, but not one that we can derive energy from. Instead, we can derive energy from starch. And if we look at this, it looks so similar to cellulose. But instead, we have 1,4 glycosidic linkages in amylose, where it's just a long straight chain. We've looked at this already, and it makes kind of a coiled structure. Or amylopectin uses 1,4 glycosidic linkages, but also 1,6, where it's connecting with a different carbon, the 6th carbon, and that ends up giving us the ability to make some branches. In doing this, we have a branched molecule that we can still digest with an enzyme that we have called amylase. And again, we'll talk about enzymes coming up, but while we cannot digest cellulose, we can digest starch. The difference is between the alpha and the beta glycosidic linkages. Here's our 2 types of starch, right? Amylose and amylopectin. And I've gone ahead and replaced the diagram of the individual molecules with a depiction you'll often see with biological molecules that emphasizes their overall shape, right? These are coils. And for amylose, it's coiled and it's just a single long coil. Amylopectin is coiled but also branched and linking. And if we look at these, take a pause here and just think, which of these two do we think is going to be easier to digest, right? Which one do we think is going to be easier for the body to break down? Think about that and then go ahead and unpause. All right, which of these is going to digest more quickly? It's going to be the amylopectin. The branching is going to help with the digestion because there's more ends where the digestion can begin. When we have molecules, it is always molecules bumping against each other, crowding around, having to find the enzyme. I mean, there is a lot of this that's just in the nature of all the million things that are going on in our body. Having more sites where digestion can begin from the ends means amylopectin is a bit easier to derive energy out of. Again, these are both starch. This is for energy storage within plants. We, however, will ingest a lot of starch from plants in the foods that we eat, so amylose and amylopectin will be coming in and we can get glucose out of them. What we use for storage is called glycogen, and this is the major form of polysaccharide storage. It really looks a lot like amylopectin, but it differs from the fact that it has this facilitating protein in the middle, and so if you look up structures, it'll look something more like this. Glycogen is highly branched. There's many ends available for hydrolysis, which means that it's going to be a very convenient energy storage for us to keep our glucose monomer units in. And in the center, right, is this protein. We see this ribbon structure of a protein. We'll see this again, and this is really to show that this is a larger molecule where instead of focusing on the individual atoms that are making up this molecule, we really care about its overall shape. And when we look at glycogen, this protein lives in the middle.